Okay, so good morning, everyone, and welcome to the Center for Theoretical and Computational Physics weekly seminar. Today, it is a pleasure to welcome Amin Doostmohammadi from the Niels Bohr Institute at the University of Copenhagen in Denmark. A few words on Amin's background. He did his first degree in mechanical engineering at the University of Tehran in Iran. This was followed by a PhD at the University of Notre Dame in the US. He was then a postdoc at the University of Oxford in Julia Yeoman's group for some time, after which he took up his present position as assistant professor in the Niels Bohr Institute in Copenhagen. And today he was kind enough to agree to tell us about his work on taming active matter from ordered defects to autonomous shells. Before I hand over to Amin for the seminar proper, just a couple of announcements from me. First, this seminar is being recorded, so by staying on, you're giving permission to your voice and image to be recorded and stored. Second, in the interests of internet stability, can I ask all of you, except the speaker, of course, to mute your microphones and switch off your cameras during the seminar. If you wish to ask a question, you are, of course, free to unmute yourself and switch on your camera if you so desire at any point. So without further ado, it's over to Amin for the truly interesting stuff.
Thanks a lot, Paolo. It's a great pleasure to talk about some aspects of my recent research. And as the title suggests, I will be mainly talking about taming active matter, but I will start by actually giving a little bit of a general overview about the active matter and how physics in principle can contribute to understanding some of biological problems. So to me, a very good example of active matter are these swarms of bacteria or this collection of cells in a tissue. Here you can see the defining features of active matter. Each bacteria or each cell that is building the material is capable of taking chemical energy from environment and converting that into motion. So they work like a machine and they locally inject energy into the system such that the whole system is constantly driven out of thermodynamic equilibrium. And as a result of this local energy injection, the system is capable of showing patterns on the scale that is much larger than the scale of the individual particles, as clear from these vortices or jets of flow in the system that, for example, in the bacterial system could be on the scale of 50 to 100 micron, while the size of the bacteria is 1 micron. What is fascinating about this is that we can go even one scale down. We can go and look inside one of these cells, and the materials that build the cytoskeleton of these cells, the filaments that are put in motion by motor proteins, they are also active. They have the same properties. They locally inject energy, they drive the whole cell out of thermodynamic equilibrium, and they drive essentially the motion of the cell. Now, what is interesting about these natural systems is that if we understand what's going on in terms of this collective organization, we can also get ideas for designing our own materials that are capable of these self-organization properties. A very successful example has been the mixture of microtubule filaments that are put in motion by kinetic motor walking in between. And if you put many of these filaments together, then we have again a material that shows these properties. It is driven locally, it is capable of creating this collective motion, but it is much simpler than the cells or bacteria that I showed. It has much less chemical complexity, it doesn't have DNA, and so on and so forth. So we can start asking interesting questions about control of these materials. For example, what happens if we put it in a confinement? And it turns out actually if you put this in a rectangular confinement, that chaotically moving flow self-organizes into a one shear flow. One can make this even more interesting and put it in a racetrack, and then the material kind of starts to flow on its own. And the amazing thing is that it can reach speeds of about millimeter per second and can persist up to meter scale, which is comparable to the current microfluidic technology. This is very interesting, and it is also interesting that it is generic. It also happens, turns out that it happens also for bacteria or cells. If they are put in the confinement, they can create this coherent flow. Now, with this very brief overview of some of the systems that we now categorize as active matter, I would like to give a big picture about why we are studying this that is also relevant to the structure of this talk. At one part, I think the goal of these studies is that we use ideas from physics to understand something about biology of the living systems. For example, we know now in humans, when cancer invades, in more than 90% of the time, it does it collectively. So it's important to understand why it does so and how it does so, how the cells coordinate their motion to create this kind of collective flow. The other fascinating example is during the organ development. Here you can see the fly embryo at the early stages of formation. Each dot shows one cell. And you'll notice that after a while, these cells, they coordinate their motion, they create this large scale flow, and then they self-organize to these very neat stripy patterns. And then each of these stripes would go on and form different parts of the fly body. Again, the idea is that if from the physics understanding, we can learn something about how these self-organization problems arise. But equally important is also to be able to learn from this natural system and imitate these materials in order to build our own materials that are capable of self-popping and self-organization. For example, these living vesicles have been shown to exist experimentally, and also these there has been ideas about suspending gears inside bacterial baths and extracting some mechanical work. So based on these two broad categories, the structure of this talk is going to follow the same idea. I will first start by giving you two examples of how we can use ideas from physics to learn something about biological functionality. And then I will focus about now how we can use this understanding in order to control and maybe design our own materials. The first example I want to talk about is on bacteria. These are Pseudomonas bacteria. They're very infectious. I normally have a picture of them here, the picture of the infection, but I figured it is right before lunch there, so that's not a good idea. So I leave that to your imagination for now. But these bacteria, these are highly infectious, and they're capable of moving by these structures called pili. They're essentially feet that they use in order to walk on surfaces. So what our collaborators in Oxford Zoology and in Sheffield did was to create a mutant of this bacteria that essentially has more leg and is able to move 2 times faster. So we have a regular bacteria and we have a mutant that individually is capable of moving 2 times faster. Now, the question is that what happens when we look at collection of these bacteria? Is moving fast giving the bacteria some advantage in terms of also invading space or not? So this was the original question. So, and here you can see kind of a racetrack experiments. On top, you have the slow-moving normal bacteria. On the bottom, that's the mutant that individually moves 2 times faster. But to our surprise, although initially the fast one took over a long time, always the slow bacteria was able to win the race. So somehow, these slow-moving bacteria were able to outcompete the fast-moving mutants when they move collectively. And we wanted to understand why this was happening. Indeed, we observed the same pattern also in the mixture. So that was the racetrack, they were separate, now we can mix the fast and slow one. Now the fast ones are marked by these purple GFPs. And again, the same thing happens. Over time, we can see the front getting dominated by the slow-moving one, while the fast-moving bacteria, they get stuck in behind. And looking more closely, we see that they actually get stuck in some kind of structure that we call rosettes. So essentially, what happens is that moving individually faster results to formation of these rosette structures, but at this point, we don't know why they are moved, but why they are formed. And because they get stuck in these rosettes, they cannot invade the new territory as efficiently as the slow-moving wild-type bacteria. So if we understand why these rosettes form, then we have an explanation for why the fast-moving bacteria are not able to invade effectively as a collective. So in order to understand why these rosettes form, we started looking more closely at the structure of this system. And we characterized the orientation field. Immediately you can see, so different colors are showing different orientations. Immediately one can see formation of domains with the same orientation. But more importantly, at the points where the domains of different orientation meet, then we have singularities where the orientation is not defined. And these are known as topological defects. Apologies to the experts here. I'll just do a very quick overview of the defects. In a system, in a nematic system that has a head-tail symmetry, the lowest order defects that we can have are these specific shapes called that have shape of a comet or trefoil. And we assign to this certain charge. So this one is a plus half. That is simply because around the singular point, if I follow a 2 pi rotation, the alignment of the particle rotates only by pi. So that one has a charge plus half, while the opposite happens here. Taking a 2 pi rotation in anti-clockwise direction, the orientation rotates by pi in a clockwise direction. So we have minus pi over 2 pi, so minus half charge. And one can have also higher order, like asters or vortices, and these are not specific to this bacterial system. They are essentially in any system with orientational order. When the order breaks down, we're going to have these topological defects, as we have in fingerprints or on the back of the head. So the fact that we observe these forms of singularities, these topological defects in a bacterial system, indicates that they have this dominant nematic symmetry. But what separates these systems from the passive counterparts is that things like that these topological defects are continuously created and annihilated. You can see examples here that plus half, minus half, they get nucleated in pair, they move around, they find maybe another defect of opposite charge and they annihilate, but at the steady state you have a certain number of topological defects in the system. We started by thinking of a simplest possible model to look at this system, and that was a system of self-propelled rods. These are essentially a bunch of rods interacting only via volume exclusion. There is no rules for alignment, there is no attraction. It is just a Yukawa potential to result in volume exclusion, and each rod has a force on it. So set of Langevin equations describe a system, and qualitatively they seem to do the same thing. So they move around, they form these plus minus half topological defects, they get nucleated in pair, annihilated in pair. But we can do better. We go and we compare the velocity field, the average velocity field around these topological defects between the experiments and this discrete model. And here immediately one can see the difference between these two defect types. The minus half, this is threefold symmetric and has also symmetric flow structure around, while the plus half has already a broken symmetry in its shape, and indeed the flow around it also has a broken symmetry, and the defect starts moving towards its head, as it is evident from the average flow around it. We also asked if we can compare it to the continuum theories, and the continuum theories to explain these systems are essentially the equations of liquid crystals that have extra contribution to make it active. So in these systems, we need to, our order parameter is essentially this nematic orientational order that is described by the nematic tensor Q. It has a time derivative and advection. It relaxes to the minimum of a free energy that would try to penalize deformations in the alignment. So this is like an orientational elasticity. But what's important is that in addition to just getting advected with the flow, if we have elongated particle, they need to also respond to gradients in the flow. If there is vorticity or if it is shear, they need to respond accordingly. And that is expressed by this co-rotation term that characterizes the response to the symmetric and anti-symmetric part of velocity gradient that are the strain rate and vorticity respectively. Now, these orientational dynamics is then coupled to the mass and momentum conservation. Without these active terms, these are essentially equations of passive liquid crystal. But we know that these bacterial systems are active, and the effect of activity to the lowest order to stress can be expressed by being proportional to the nematic tensor. One can derive this mathematically by considering each of these creating stressless and then coarse graining. But physically, all this means is that, so this is stress, the divergence of stress gives force. So any gradients in the nematic order are going to create force and correspondingly create flow. But the advantages with this continuum description is that we can basically prescribe the shape of a defect. So our Q is known, and then we can solve for the velocity using Green function. And we do that, the analytical calculation actually matches quite nicely with both the discrete model measurements and also the experiments, and shows again the difference between a minus and plus. Now, what's important is that then we can use this theory. Based on the theory, we expect that these plus half defects that have their kind of own self-propulsion, we expect them to be faster for the faster moving bacteria. And this is indeed the case in the experiment. So here you can see the mean square displacement for the normal and the wild type, while the minus halves, because of the symmetry in their float, they are basically moving around diffusively in the system. The plus halves, they have this ballistic motion, and also the plus half for the fast moving mutants are much faster compared to their wild type counterpart. So what does it mean? At this point, then we had the idea that actually what happens maybe in this system is that the plus half defects, these comet shaped defects in the fast moving bacteria, they are faster. They can basically come very close to each other and form a virtual +1 defect. And we know from the theory of liquid crystal that these +1 in nematic has very high energy that cannot be sustained, and the phenomena of escape to the third dimension should occur. So once these defects come close to each other, they form this virtual +1, and then the bacteria in between, they are basically pushed out, so they stand up on their end. We can check this in the simulations with the rods. We can basically shoot two defects in a fast-moving bacteria to each other. We check that in slow-moving bacteria, they basically go past each other, but in the fast-moving one, then some kind of a reverse domino effect occurs. Everything stands up. But more importantly, we see the same thing over and over in the experiments. Exactly when these two defects come together, the bacteria get out of focus, showing that they are standing up on their tail. And the same thing happens also in the mixture. As you can see here, that the fast-moving ones are the ones that they stand up when the two defects come together. So this is interesting because it kind of suggests that evolution might have put some speed limit on how fast these bacteria can move. And that speed limit is basically because they are following the constraints set by the physics of active liquid crystal. So if they move faster than certain velocities, then they get stuck in the structures of their own creation. Now, the second example that I wanted to give you about using ideas from physics in biology deals with a completely different system. I talked about bacteria. Now I want to talk about cellular tissues. These are epithelial cells. These are the cells lining surfaces and cavities in our body. But in these systems, we can also identify these topological defects based on the elongation of the cells. And in the experiment, we find that the cells actually also get expelled at the head portion of these +½ topological defects. And the experiments show that this happens in kidney cells, human skin cells, and both aggressive and non-aggressive breast cancers. So there should be something generic going on. You can see an example of this. If you look under the microscope, normally these white spots are where this extrusion happens. And this is quite important for keeping the integrity of the tissue in general, because cells are constantly dividing and they need to also at the same time get rid of some of the excess cells. And you can see example of this extrusion happening at the head of a +½ defect here. So again, we wanted to understand why this happens at the head portion of these +½ defects. So we started comparing the shear rate and velocity field from the continuum theory to the experiments. They work well, but more importantly, in these experiments, we have access to the mechanical stresses that these cells exert on their underlying substrate. So we can compare the isotropic stress around these defects. Why isotropic stress? Because the isotropic stress tells us how much compression or tension we have. So the positive ones, these are the tensile stresses, and the negative regions, they are the compressive stresses. And immediately one can see both in experiments and continuum model that at the head portion of these +½ defects, we have these strong compressive stresses. And we think that these compression is enough to basically push the cell out of the monolayer. And indeed, in the experiments, we can see build-up of these negative compressive stress before the cell is being pushed out. So we reformulated how this process of extrusion happens. Consider epithelia as an active system. So this system creates pairs of topological defects. There's a certain density of these defects. If the cell happens to be there, we initially thought that the compression basically pushes it out, but further studies, we actually found out a little bit more complicated than that. The compression actually is capable of affecting a certain mechanotransducive pathways inside the cell, and lead to the cell death. So the compression first kills the cell, the dead cell loses contact with the surrounding and almost immediately is pushed out of the monolayer. But if you're thinking about this epithelial system, one can immediately notice a significant difference compared with the bacterial system. The bacteria have a fixed shape. They are rod-like. These cells, most of them are actually circular. And it is interesting that because of the shape changes that they create, they are capable of showing these nematic order and also showing these topological defects. So we wanted to understand this further. Again, we came to a model, the simplest model we could think of. So this is a model of the cell tissue, just thinking about them as collection of droplets, but not regular droplets, but active droplets. And to do this, we use phase field formulation. Essentially, this follows the regular, this phase field is defined such that it has value 1 inside the cell and 0 outside. And then we have one phase field for each of the cells. And it follows without, if you forget about this term, it's essentially the Can-Allen equation. The relaxation towards minimum of the free energy, this gives us the properties of the interface, such as surface tension, and we can include results of the details of cell repulsion. But more importantly, we can make this active by considering this extra advection term, where the velocity is determined from the force balance. So these are overdamped. We consider this as an overdamped system. So velocity is related to the force. So all we need to make this system active is to define the correct stress. And we started by, again, the simplest form of the stress we could think of. The idea was that because these cells are constantly changing shape and activity should be proportional to how elongated or how deformed these cells are, we consider the active stress to be just proportional to the shape tensor of the cell. So the largest, the eigenvector corresponding to the largest eigenvalue of this shape tensor essentially gives the direction of the cell elongation. And all these, and what this stress means is essentially if you have a little bit of fluctuation and the cell is a bit elongated, this will amplify. This was the simplest that we could think of. More recently, we have done also comparing with experiments and looking at the more sophisticated ways of putting these stresses in. But I think for the purpose of this talk, let's just focus on this very simple one. So essentially just one parameter here would drive the dynamics of the system. And indeed, again, qualitatively, one can see that this simple form of active stress can result in the whole system pushed out of equilibrium, and we can see the vortices. But more importantly, we can compare now this average isotropic stress around the topological defects from this discrete model to the continuum model and to the experiments and get satisfactory results. So essentially, these high levels of mechanical stress at the topological defects are observed both from the continuum models, from this discrete model, and from the experiments, and we think these are responsible for killing the cells and pushing them out. And examples of defects and their biological functionalities are now found in many systems. But the important point that I want to make at the end of this section is that I have shown examples from two different systems, bacteria and epithelial cells. And I have compared this with a discrete model for bacteria and a completely different discrete model for cell, and also compared it with continuum model of active nematics, and they all seem to agree. So that is also the beauty of the physics, that the physics theories and physics ideas are generic and can be applied to different systems. So for the rest of the talk, I'm gonna focus on this continuum theory that at least seems to be relevant for these bacteria or cellular systems. And so far, I hope that I have convinced you that these defects are important, but we have a problem with them, and that's the fact that these have very chaotic trajectories. Indeed, in many of these biological systems, they are interleaved with these chaotic flows that are known as active turbulence. That's an interesting topic of research on its own. Again, I will not talk about that today. Instead, for the rest of the talk, I want to focus on how we can tame this otherwise chaotic flow in active matter, and I will focus on the theory part here. Indeed, the continuum equations that I showed, if we simulate them, we observe patterns like this. So we have these chaotic flows. One can, by eye, even see these certain lengths of the vortices that appear in the system. So there is some characteristic lengths in the system that is driven by the activity, and it's accompanied by the chaotic motion of these. The first idea we had was that what if we introduce friction to the system? Because we know many of the biological systems, they actually live in contact with substrate where there is actually some form of friction. What friction does is that it introduces a hydrodynamic screening length. The larger the friction, the screening length becomes smaller. So we expect that as we increase the friction further and further, this screening length becomes smaller and smaller, and at some point should become comparable to the scale of these vortices. And indeed, that is when interesting things happen. This chaotically moving flow self-organizes into a lattice of vortices. And more importantly, this lattice is interleaved with the ordered structure of topological defects that have some kind of crystal structure. But the important point is that this happens only when this screening length becomes comparable to the intrinsic active length scale of the flow. But friction is not the only way of introducing hydrodynamic screening. We can also do that by putting these things under confinement. So here, when we look at numerical simulations of the system, put under this confinement. So we have no slip walls on top and bottom and periodic boundaries on the sides, and either increase activity or height. They show similar effects. So for small channels or small activities, the whole flow self-organizes into one coherent structure that moves. It breaks the symmetry and chooses either right or left with the same probabilities. At larger channel heights or larger activities, we recover the active turbulence state. But again, when the screening length becomes comparable to the intrinsic length scale of the vortices, then we observe this beautiful vortex ordering. What's different about this vortex ordering is that it leads to some kind of dynamically ordered structure of topological defects. Here, the plus half defects are shown by green and the minus half by pink. The minus half, as I showed earlier, based on the velocities, they don't have their own self-propulsion. They interact elastically with the boundaries, so they get basically pushed to the boundaries, while the plus half, they interact with the flow. And if you follow one of these, then you can see that these two defects, they interact with one vortex, then they move to another one, and by doing so, span the entire system. And this reminded of us of something that is happening in Scotland quite a lot, and that's this.
Our next dance is one you'll definitely know. Strip to Willow, also known as Drops of Brandy. It's a real favourite for both Cayley and Scottish country dancers. The first couple turn by the right hand at the top, usually a barrel to Cayley's. The first lady turns second man left hand, her partner right.
You get the idea. So based on this, we call this a Cayley dancing stage, because essentially these defects, they dance with one vortex, move to another one, and by doing so span the same system. And interestingly, then this was actually found also in the experiments from Francesca Guest's group, that confining microtubules also shows this Cayley dancing of defects. And also there was a beautiful work from the Lisbon group that also showed that this also has consequences in terms of the active-passive interfaces in a channel confinement and creating these dancing states of the defect. But in addition to being a acute state, this vortex lattice stage also is an intermediate state before going to turbulence. In fact, if we increase activity a little bit from this vortex lattice state before going to the turbulence state, we can have a coexistence of ordered vortices and disordered vortices. We can quantify this by measuring the entropy, which is just a fancy word for the magnitude of the square of the vorticity. So we can average it over the channel height. Then we get very nice one-dimensional signal, which is ordered for the vortex lattice and disordered for the turbulence. Now, this is only for one time point. I can now take this one-dimensional signal and look at it over time. Then I have this chemograph, which is essentially a background of vortex lattices. And in between, we can see this, what we call path of disordered flows that are generated. What's amazing is that these paths, they can start, they can branch out, and they can die out, and so on. Now, interestingly, we see that for low activity or small height, these perturbations or these paths, they die over time. At high activities, they go to the entire system. But there are some critical activities where the rate of branching and rate of decaying, they balance each other. So we always have a certain fraction of our channel occupied by these disordered flows. We call this turbulent fraction, and we can measure it as a function of this activity parameter, which is essentially not dimensionless activity. And then we can see that it follows a power law growth with exponent 0.276. Why is this important? Because this is exactly the exponent for the directed percolation. And what is even more interesting is that for the classic inertial turbulence there were 2 independent studies in 2015 showing at the Couette flow and channel flow, and showing that the classic turbulence belongs to the directed percolation universality class. And what we see here is that this essentially 0 Reynolds number active turbulence, at least in a channel, also seem to belong to the same universality class, which to me is quite interesting. Now, this was in 2D. We can do the same practice also in 3D. Now, we confine the active nematics inside the 3D channel, and we play with the height of the channel in the 3rd dimension. For small heights, we recover essentially a quasi 2D system. So if we look at each plane, it is similar across the z direction, both in terms of the velocity field and also in terms of the director. And since it is similar, then those point defects that I had, now they become straight lines, and the system moves around. So it is a quasi 2D active turbulence without any 3D flows or any 3D features. Now, if we increase the height of the channel or we increase the activity, actually it becomes more interesting. And these defect lines, they start to buckle and they start forming loops. And the velocity field also becomes 3D. We find indeed that this transition from 2D to 3D happens exactly when the curvature of the defect line changes and they start buckling. And indeed, we find that it happens because of the emergence of twist in the system. So in 2D, you cannot have a twist, but in 3D, when the activity is high enough or the height of the channel is high enough, then the twist instability kicks in that starts twisting these topological defects, and that buckles the defects and results in transition to 3D. And these defect loops also were observed recently in the experiments on microtubules that were stabilized by FD viruses. And they also observed these twists and bends. Now, another way of controlling these active materials is by putting them in contact with some pre-patterned surfaces. And this is work in collaboration with Oleg Ravrantovich and Igor Aronson, who had this brilliant idea on living liquid crystals. So essentially, they put the bacteria inside a passive liquid crystal. The advantage is that the direction of the alignment of this passive liquid crystal can be controlled externally. So that gives us some control over the alignment of the bacteria, but it doesn't restrict their motion. What we started to do was to then put these bacteria inside this sort of bend display structure. So this C shape, so we have bend here and display here. And one can immediately see that these bacteria, they get condensated into these display regions. One can see an example from movie. You put the bacteria, they immediately move towards display region, and the concentration increases and increases. Instabilities kick in, but the instabilities cannot grow. They basically, the things are moving, they form these bend-like instabilities, but they cannot grow, and they continue forming these strong jets. From experimental perspective, these are interesting because then you can show these jets are strong enough to basically transport micron-sized cargos, and even 6 of them. But again, we wanted to find a way of modeling this system and understand this process. So we went back to the continuum model. Now, in addition to the orientational order and the mass and momentum conservation, we need to also introduce the binary order parameter that distinguishes between the active and inactive regions. And to account for this alignment with the pre-patterned surface, then we consider additional contribution to the free energy. So we can prescribe the director field, the nematic tensor for the background passive liquid crystal, and we can introduce the energy penalty for deviation of the active material from this prescribed orientation. So we can do the same kind of experiments. We can start from slab of active matter, put in the C-shaped geometry, and we can see immediate this condensation into the display region. And can also compare the concentration profile with the experiments, but more importantly, we can also observe the formation of these undulations in the stream that they cannot grow above the certain length that is set by the length of the bend structure that is put in the system, and can find the relevant parameters that can lead to these stable streams in the system. And I would like to finish this taming of active matter, but by one that I find actually most interesting, and that's using topological constraint. So this system of the microtubule motor protein mixtures that we saw at the beginning, one can also make emulsion of this at the oil-water interface. And then it behaves astonishingly like a living organism or living thing. It moves around. As long as ATP is available, it moves around. It creates these beautiful protrusions and dynamic patterns. And it is amazing because it is alive, but it doesn't have any DNA. It is not really chemically as complicated as other systems. But can imagine that modeling this kind of system would be a nightmare, because one needs to account for this 3D shape, one needs to account for these deformations and formation of protrusions, hydrodynamic interactions of and the interactions between the active constituents. But we recently found a way of doing it. Essentially, in a nutshell, what we do is that instead of having the binary order parameter stabilize in one phase versus the other, what we do is that we design the free energy such that the nematic order is stabilized only at the interface. So the inside and outside are isotropic. Only at the interface we have the active nematics, so that forms an active nematic shell, and it generates these very beautiful patterns. It can move around. It can you can clearly see again minus half defects and the plus half defects that move towards these protrusions and create these dynamic shapes. But based on this, then we can observe that activity actually can lead to formation of very various shapes, some of them very stable, some of them more unstable. And depending on the magnitude and the type of the activity, you can have some control over the morphology of the system. And also interestingly, essentially now I have come back to where I started. We found that if we consider a hemispherical shell of these active materials, then when the two plus half defects are formed and come together, they can form a plus one and create protrusions in the system. And indeed, we can find that there is some correlation between these positive and negative defects and the curvature of the system. So basically, once the plus halves are formed and they form a plus one and move and form the protrusion, another minus half can be left behind and leave in the valley. And interestingly, this was recently observed also in terms of the morphogenesis. So these are, the system is a sea animal called Hydra. It is very interesting because if you cut any part of it, it regenerates to the original shape, as you can see here. For example, you take a fragment and then put it under microscope and wait around 40 hours or so, you can see that the stars basically first elongating, then forming these protrusions, forming mouth and feet, and it's essentially going back to the original shape. But the interesting thing is that when they mapped the actin skeleton of this animal, then they found the formation of these topological defects, essentially governing the formation of mouth, foot, minus half being left behind at the valleys, and then also 2 plus half coming together and forming the tentacles and forming a plus one defect. So with that, I just come to the end. As I mentioned, the idea that I wanted to convey with the talk is that we can use the physics to understand something about biology, but also we can learn from this system to design the materials and to control them. And we are working on this from the theory side and also trying to do some experiments on these systems. And also the experimental systems are starting to show some promise in terms of designing these sort of shape shifting materials. But the nature is amazing at self-organization. We are not, but we are learning quite a lot. Thank you very much for your attention.
Hello, thank you. Thanks very much to Amin for a truly fascinating seminar. And questions and comments are now invited. So who wants to be first? Who has questions, comments? Oh, I see something here. Elio Figueiredo, please ask your question.
Yes. Hello. First of all, thank you for the presentation. That was very interesting. How the emergent properties of these fluids, and I was particularly surprised by the slow bacteria just completely overtaking the fast bacteria. I was definitely not not expecting that, or at least not in the way that it actually happened with the faster ones standing up. My question is a bit, perhaps a bit outside the scope of the work, but theoretically, since the defects result in bacteria standing up, bacteria or other particles just standing up, would it be conceptually possible to insert material in a fluid in such a way that it would coalesce and bring out specific, I want to say contaminants, but not necessarily contaminants, just particles within a fluid, and then that it could easily extract, because it would go out, seek them, form those defects, and then bring the contaminant, the particles that you're interested in, to the surface, so you could easily extract them. So no need for filters or anything like that. Do you think that would be conceptually possible?
That's an interesting point. I think that's more on the practical side, and it's certainly interesting. Yeah, I think for example, one thing that I can think about is that if you do something like these living liquid crystals, if you then mix this with this liquid passive liquid crystal, and you have some control over these passive ones, so you can impose certain topological defect structures in the system, and then you can basically then separate the fast ones in this way, that they get basically accumulated in certain defects and they stand up. And then if you have some microfluidic device on top, that it can wash away the ones that have come up. Yeah, that could be interesting. Also, the other thing is that, so in these experiments, the things basically, when they come up, they stand up because there is an agar surface on top. If you relax this, I think it's also possible that they move on top and form another layer. And then they can also divide and create their own layer. Yeah, but I don't know if that answers your question, but I think it should be possible to come up with some smart design principles to make use of this sort of physics in the system.
All right, thank you.
Okay, we now have a question from Rodrigo.
Amin, thank you for this extremely interesting seminar. I learned a lot. And I would like to ask about, in the beginning of your seminar, you showed these two different bacteria with different velocities, and they form these Rosette forms and regions. If there is a way to predict for which velocity or which parameters it will happen, because we could not model these using continuum models, I think. And how do we know until each region or of parameters we can use continuum models to simulate interface in bacteria? Like
yeah, that's an interesting question. So in terms of the self-propelled model, so I didn't talk about it here, but in the paper we actually did look at how these sort of rosette formation happens as a function of the force that we put on the bacteria. And there is a certain threshold in terms of the force that these bacteria have. And we can find that from the self-propelled rod model. From the continuum, there is extra complexity that we did not include in this continuum model, and I think it is also something that is missing in several continuum models, is that in addition to this nematic symmetry, the bacteria are also polar, right? For example, in the self-proper rod, actually, they are certainly polar. They have a force on themselves, while when you do the nematic modeling, you do not consider this effect. So I think we have recently found a way of doing this. So essentially combining the nematic and polar thing to get both half integer and full integer defects. But I think in order to answer this question, really needs to account for this polarity in order to really find a threshold. But I think once we have the correct set of equations that also include this important aspect, then that should be possible to gain some insight based on what is the threshold that needs to be passed. But I think there's some kind of energy barrier also to form these sort of Rosette structures if you allow your continuum director field to also escape to the third dimension.
Thank you for checking the paper.
Okay, thank you. Nuno.
I mean, thank you again for your talk. I have several questions, but one question is related with this problem of the two types of bacteria, where you have the fast and slow bacteria. Can you connect that with the result that is known, for example, for where you have this argument that slower is faster, typically when they go through constraints? I know that here you don't have constraints, but have you thought about any connection between this result and what we know there?
Well, that is a actually, when we wrote that paper, we started actually writing about pedestrians to motivate it, because we wanted to sort of, but I thought about it a little bit at that point, that, you know, when you are doing these sort of larger scale concerts and so on, that if you are able to tag people and look at the ones that are moving faster versus the ones that are less active and staying essentially at their place. But I don't know. I think the one thing is that, you know, this, at least what we find is in terms of the liquid crystal features, but when you are dealing with pedestrians, they are not really elongated objects in plane to form this kind of defect.
Yeah, but you break locally the symmetry because you have a preferential direction to move. That's why most of those studies were done with constrictions, because you ask people to go through a door or something like this, right? Yes. And I was thinking that in the case of the bacteria, they have this characteristic time for changing the preferential direction of their motion, right? So somehow they will accumulate at a certain point, and this will make obstacles for the next one. So I was, well, thinking if there could be any connection between the two results.
I think my answer would be, yeah, I can make a conjecture that there could be connections exactly from the mechanism that you mentioned in terms of the breaking of the symmetry and so on. But I cannot concretely say that until, but I don't know if I think it's called for actually a discussion, but yeah, so if I think probably then you are thinking about if we can make some connections to the density variations and so on, not in terms of the orientation, but if you have some sort of singularities, and then what would be the ramifications of that in terms of the density variations. Interesting point, Nuno. I have.
Thank you. I have another question. Is related with the branching process that you have shown, right? You said that you were surprised to see that it's still in the direct percolation universality class, right? Yes. But you are looking at low Reynolds numbers, right?
Yeah.
For example, if you have turbulence with high Reynolds numbers, you know that you have all these scaling variants and things like that. So you have long-range correlations that in principle could kill the direct percolation universality class. But what do you have in mind when you say that is surprising? Were you thinking about any mechanism of breaking the universality class when you are in low Reynolds number, where typically you have a certain characteristic length and and things are correlations are finite in terms of.
What I was thinking was that normally to get a directed percolation, you need this absorbing state, right? That is like in a classic turbulence, we have this laminar state, and then you put this pressure perturbations. And here, your absorbing state itself is dynamic, is this vortex lattice state, and the paths that are being created, they are also consequence of some driving inside the system. So it is, so in that sense, it is, yeah, so maybe actually it is more like a, I think to me, it is more closer to a directed percolation with a conjugated field or something like that. But still, to me, it was interesting that we could see exactly the same exponents and stuff starting from some dynamical state rather than a purely absorbing state. What would be interesting to me is that if the same thing happens also in the bulk. Now, if you don't have it, because here we had the confinement, and then one can argue that the confinement was also playing a role. But if also in the bulk, if you vary the friction of the system and go from the vortex lattice to the chaotic, if that is also a directed percolation, or even if there are some things in terms of the conformal invariance and so on that you would observe in this system, because that would really be an advance in terms of understanding these chaotic flows if we know what kind of universality class they actually belong to.
Yeah.
Right?
Yeah, because in 2D, there are some works that look into conformal invariance in normal turbulence.
Exactly.
Exactly. And the only way of breaking the universality class in a branching process would be through long-range correlations that decay slower than the dimension of the system. So I was not thinking in terms of Aries criterion, right? So I was not seeing why here will not be direct percolation. So that was my point.
Okay. Yeah. So you are saying that it was expected that it should belong to the directed percolation.
Except if you have a mechanism of imposing a correlation in the way the branching occurs as a function of the distance that decays algebraically. So with a power.
I mean, that's what I was saying in terms of here. Basically, the path creation in the absorbing state could itself be somehow correlated, because it is not that you are putting, like a classic turbulence, you are putting a pressure perturbation at a certain point. Here you can have several points where activity results into the formation of the path, and that was kind of different from at least my limited information on the directed percolation. But yeah. Thanks.
Thank you very much.
Yeah.
Okay. Any more questions? I don't see any more raised hands. So I think we wrap up for the day. So Amin,
yes,
thanks very much again for a truly fascinating seminar and a very lively discussion. And well, hopefully we'll meet some other time, and this time hopefully in person at some conference or something.
Yes, great. Yeah, thank you very much. I also want to thank you, and it is not often that you get these sort of lively discussions after a Zoom talk. So it was very nice to have the discussion and see familiar faces. And thank you very much for the invitation.
Okay, thank you. So recording stopped.